Luba, am I ready to start?
Yes.
I'm allowed? Okay. Thank you. Good morning, everyone. Thank you all for joining our third Biotech Connector this year. Uh, my name is Vladimir Papa. I'm the Chief Innovation Officer here at the Project National Lab for Cancer Research. Biotech Connector, an event for those who haven't been before. It's a joint effort between Frederick National Lab for Cancer Research and Frederick County Chamber of Commerce. Thank you, guys. The event is an exclusive quarterly series that brings together the local biotech community for an inside look at advances and chance to share expertise as well as opportunities to network with other scientists, entrepreneurs, and experts in the area. Frederick National Lab, it's a collaborative enterprise, as some of you know. We are a national lab that employs about 2,400 people and probably close to 1,500 subcontractors. So we do cancer research, infectious disease, and HIV and AIDS research. We are always interested in partnering, but we also have other opportunities for companies and for entities. So we have subcontracting opportunities, which are available on our website as well, or just talk to some of us. And we always have a careers page where we are hiring, excuse me, where we're hiring a lot of folks at all times. So today we have actually an amazing lineup of speakers who will be presenting on the topic of structural biology and its application in biomedical research. But before I move onto speakers, I would like to cover some of the logistics. I think there should be a next slide. If we can, for the folks that have questions for the speakers, we'll be moving through the speakers and we'll have time for a question or two after each speaker. If you're in the room, just stand up, state your name and ask your question. If you're online, for folks, and we have a lot of people online, please either unmute yourself or just put your question in the chat. During the presentations, for the folks that are online, if you can remain muted, that's great. And let me introduce the speakers now. Like I said, we have three amazing speakers. The first one is Dr. Ruth Nussinov. She's a senior investigator and the head of the Computational Structural Biology section here at the Frederick National Lab for Cancer Research. Dr. Nussinov has obtained her degrees from University of Washington and Rutgers. Her dissertation developed the first dynamic algorithm for folding of RNA, which is still taught in bioinformatics classes in US and Europe. She was a postdoctoral fellow and research associate in Weizmann Institute, Berkeley, Harvard, and the Computer Science in Tel Aviv University. She joined the Sackler School of Medicine in Tel Aviv University, where she was a full professor and became affiliated with the Frederick National Lab. She was elected as a fellow in several societies, won multiple domestic and international awards, and served in several roles in academic community. Our second speaker for today is Dr. Carter Mitchell. He's a protein chemist and structural biologist with over 20 years of experience isolating, characterizing proteins from recombinant and natural sources. Throughout his career, he has been exposed to a range of structural and biomedical techniques and developed interest in the development and manufacture of bioproducts. As a graduate, he enzymatically and structurally characterized multi-domain proteins involved in metabolite biosynthesis, and then also led a drug discovery pipeline with a team of scientists that isolated and characterized bioactive small molecules, peptides, and proteins from mammalian microbiome and marine aqueous. Currently, Dr. Mitchell leads the scientific team at Kemp Proteins here in Frederick. Our third speaker is Dr. Manu Kohli. Manu Kohli, I apologize. He's a principal scientist, scientific advisor at Charles River Labs. Dr. Kohli has over 20 years of biotechnology and pharmaceutical experience with expertise in target identification, validation, drug discovery, and development. He has a variety of disease he has led senior scientific research teams in the biotech and pharma sector to advance drugs for a variety of disease indications. Dr. Kohli has previously headed research programs in small molecule immunotherapies, biologics, and cell therapies, and has developed research strategies using diverse modalities. Dr. Kohli has a Ph.D. in biochemistry and molecular biology from Georgetown University with postdoctoral training from Johns Hopkins University. He has published many research articles in top-tier scientific journals and holds numerous patents. Now, I would like to welcome our first speaker, Dr. Nussinov.
So thank you very much for the invitation to speak here. It's the first time we come here. So where is my presentation? Do I simply go straight? Yep. Okay. And to blow it up, it already shows. Okay. So as you can see, I changed the topic or the title of my talk to something that we are very interested and are working on right now. And this is figuring out mechanisms in oncogenic mechanisms. Within this framework, we have been particularly focusing on the RAS signaling network. It's not surprising. Actually, we started working on it in 2013, a few months before the RAS project was announced as a project of national interest. So we were right. We have a sense that this is the way to go. This is the significant way to go, and we were happy to be vindicated. So RAS, I'm going to discuss RAS, to mention RAS itself, and its major pathways, the MEP pathway and the PI3K AKT. All the proteins that I'm going to show now, we have been working on them, either worked on them, or are in the process of working on them now. So here is RAS itself and its activation, the proteins involved in its activation. Sorry, I see I'd better wear glasses. And we see, actually, I cannot, I guess there is no way for me to step away, right? Opa. Oh, okay. RAS, its activation, the proteins involved in its activation, then the dimer, RAF, MEK, ERK, and here also we have been working on the KSR with ERK. Together, these two constitute the MEPK pathway. We also looked at ABL, which is a key kinase in leukemia, and the parallel pathway, or not the parallel, I should say the complementary pathway of the PI3K AKT, which is the growth pathway. We have been working on PI3K, a lipid kinase, P10, a repressor, a suppressor of cancer, and a pathway involving PDK1, AKT, and the all-important mTOR. These two pathways, the MEPK and the PI3K, signal down, entering to the cell cycle. We have been working on the cell cycle, which has several stages, as you can see. We so far have been working on G1, which is as I say, the first step, involving the main kinase there is the CDK4 and 6 and its partner, cyclin D, and the CDK2 with cyclin E. And we are planning to go throughout. To give you a sense of the type of questions that we have been asking, as I say, we look at activation and signaling, and in this case, we ask, for example, there are these several steps, and we see that CDK4 catalyzes or acts in the G1 phase, which is the long phase, and then we look at the transition between the G1 and the S phase involving CDK2 and cyclin E. And we ask, why did Mother Nature select or optimize CDK4 and 6 for the G1 phase and CDK2 for the transition? Our hypothesis was that because the G1 is takes on a longer time scale, the transition between the G1 and the S is short. It probably has to do with the kinetics of the CDK4/6 versus CDK2. And we have been looking at it, and so far our hypothesis has been confirmed, and what we observed is consistent with experimental data. So we are interested in the mechanisms of activation and signaling. Why? First, we would like to understand, of course, and second, because it can help drug discovery and its regimen. So mechanisms of activation of the mutant protein can help in the discovery of single drugs. However, resistance is expected, unfortunately, as we all know. And the mechanisms of cell signaling can help decide drug combinations. And drug combinations lessens the resistance probability. So here, to explain the essence of the drug resistance problem. So here, on the left-hand side, we see the tumor, a section of the tumor. Cells which are marked with red circles in them, these are the most prevalent cells which have a common major driver mutations. The mutation has been presumably sequenced and is known. Among all of these, as you can see, there is one with a bluish circle. This one has a rare mutation. Chances are that when the tumor was sequenced, this cell was not identified. Now, the physician, the oncologist, give a single drug treatment targeting these red cells. What would happen is that, yes, these cells would now be decimated. We don't see them in the next stage. However, once these are decimated, the rare cell with the rare mutation can proliferate. This then requires a second drug, a subsequent drug, which once given, here shows that the tumor goes back to normal cells. In reality, this figure is for simplicity. In reality, once you give a second drug, still you don't decimate all mutant cells because there are still likely to be cells with rare mutations either pre-existing or arose during this process. So sequential drugs may encounter new mutations. So the issue is the drug combinations has a better chance. Had we given the combination a priori, chances are it would have been a better regimen. So anti-cancer drug, the key question that we have been asking ourselves is how to select small molecule combinations. Small molecules, as we know, are at the forefront of cancer therapies. Successive treatments with small molecules incur drug resistance. So the oncologists have tough choices, not just which drug to use, but also the best treatment plans based on factors like target proteins, pathways, and gene expression. We want to know those that are overexpressed or underexpressed for suppressors. So here we have combination of drugs, which to choose. And this particular slide is on the cover of a TIPS special issue on drug resistance. And this particular issue, as the editor of TIPS of the Trends in Pharmacology was working on it, and we were in touch with him, and he says, Ruth, can you submit a paper to this issue? And we were working on this particular paper, so it fit very well, and the paper was included there, actually the first paper in the issue. Still, question, how to select small molecule combinations? considering the reality of cancer's disruption of normal cellular processes, clarifies why it's crucial to understand the ins and outs of current treatment methods. For example, small molecule combinations that target multiple pathways appear a promising approach. This, however, is not the full story, because hurdles come with it. For example, pathway crosstalk. So then it overcomes the cell's own inherent regulation. And in overexpression, the same pathway combination can encounter activation of multiple oncogenic pathways by abolishing the regulation of these crosstalks, which are regulated, as I mentioned. Along these lines, strong activating mutations. Also, we have too many active conformations, because that's the issue. How many active conformations are in the cell? If too many, then that's the core of the difficulty, the heterogeneity and drug resistance. So coming back to our question, how to select the small molecule combinations? Drug combinations can target the same protein, for example, allosteric and orthosteric combinations. The same protein, I'm sorry, I should have said on top, allosteric and orthosteric, same pathway, can be upstream and downstream protein. Redundant pathways, which is signaling in parallel through same family proteins. Parallel pathways which use different family proteins. Compensatory pathways. And the issue is that combinations of target proteins in redundant, parallel, and compensatory pathways consider mutations and cross-linkages between the pathways. And overexpression contributes dominantly to the number of active molecules. Combinations should include transcription factors and epigenetic modulators. And we know that in chemotherapy, this indeed is the case, but it is not the case a priori when the oncologist prescribes drugs. So we believe that it should be combinations, and this particular combination should not be applied over a long time, because again, resistance would arise, but they should keep rotating. So bypass pathways in drug resistance, here again, RAS, MEPK, and the PI3K, AKT, and the EGFR, which acts to activate RAS. Here are the other receptor tyrosine kinases, which are not working in this case. Now, here there is a mutation in the EGFR, and we then drug it. We can drug it by even combination. We can either choose one of these or by combination. And then this pathway would be abolished. And what would then happen is that other pathways can take over, here using different RTK, different receptor tyrosine kinase. So this is bypass pathways. Here, resistant bypassing an inhibited kinase. This is an earlier work from 2017, again in the trends. So we can see the MEPK, and here there is an inhibitor to RAF, which is mutated here. Therefore, inhibition of MEK, parallel pathway with MEK, so this, I'm sorry, with MEK5, so here MEK1, 2, here MEK5 can take over. And this here now, the pathways, again, how to select small molecule combinations and the different types of pathways that I discussed. So same protein, two drugs, orthosteric and allosteric, can be same pathway, upstream and downstream, can be here parallel pathways involving proteins from different families, and here redundant pathways, the same family, and here we have extracellular, we have here, yes, we have still a different types of pathway. Now, just to show, I discussed signaling. I guess it's time for me. Okay, I'll continue. It's time.
Maybe 1-2 more minutes.
1 minute, okay. So let me just show what about the mechanism of single proteins. I talked about pathway. We believe that one wants to know the activation mechanism. Here it is RAF. Here, activation by mutations. In this case, again, these well-known mutation leading to melanoma. So exactly how it works. At the top is the membrane. This is RAS right here, the cysteine-rich domain, the RAS binding domain to RAF, and it's a dimer. And this is the RAF, the kinase domain, and it binds to the 1433 protein. And with acknowledgement, I have more slides with, again, showing mechanisms of single molecules, but I'll stop here. And thank you very much.
Any questions?
Maybe a naive question, but is there any drugs that have multiple targets, single drug that can multiple target? So I could see advantages there.
Yes, of course there are drugs that have of course there are drugs that have multiple targets. The issue is, from what I understand, I'm not a drug developer, but the whole point is that you try not to have this type of drug, because this type of drug would lead to toxicity. Because every single target that you hit eventually has some chance of toxicity. For multiple drugs, of course, we know there are toxicity issue. Toxicity issue is a major issue. We know. So usually what drug developers try to do is to have it specific. And not by increasing the concentration, right? Increasing. This is the major problem with drugs to kinases, right? Because the active sites are so similar to each other. And this is why allosteric drugs are so much, in principle, better, because they have lower chance. They may still have toxicity, but they have lower chance of toxicity because their binding sites are likely to be different, not optimized by nature to the specific catalytic action of that particular kinase. But this is a very, very relevant issue at the heart of drug development. Specificity, we know drug specificity. Yes.
So I have a question. So are there any strategies to prevent new mutations while you design the small molecules.
Excuse me, to prevent what?
To prevent new mutations coming up. Are there any strategies that you can have to prevent new mutations while you have a small molecule or antibody drug conjugates or any drug discovery?
Did you say to? I didn't still understand to prevent what? Did you say degradation?
The new mutations.
Ubiquination?
No, mutations.
Mutation.
Mutations.
Oh, mutation. Not that I know of.
Okay.
You know, mutations just occur. We wish we knew exactly the origin of the mutations, right? So what is the rationale here? I said, the oncologist needs to know which combination. So the whole idea, I should have emphasized it, at the core of our thinking is, what are the likely proteins and the likely pathways where the mutations would arise? Okay, if you know that or have a good prediction, then you are on a good path. And this is here. So that's why we try to look at this and to see, and then look what actually happens. And we have a postdoc in the group, Bengi Yavuz, who is developing a strategy of accounting, looking at what's happening and improving the chance of the selection. Yeah. So this is a computational approach based on available data in the literature databases, etc. The mutations, the frequencies of the mutations, in particular, cancer-specific, right? Cancer-specific, yeah, mutations. That's the whole thing, because so far I showed, okay, these are the possibilities, but the key question is, so which of these are the most likely, and what should the oncologist use? So the oncologist is sitting there in his office. What should he use, right? So that's the thing. And we say it's not enough. Many times it's given single drug. So single drug, again, single, because we know resistance arises, again, single drug, again, single drug. So our approach, this is not a good strategy. Yeah, much better a priori to have. But then, as I said, I kept showing which, right? So we believe that if we can come up with the likelihoods, nobody is absolutely certain, never, ever, we know that, but the likelihood, yeah. And then, of course, you want good drugs for the particular proteins, absolutely. And we know that the drug, there are some new ideas, exciting new ideas in drug discovery. I look, for example, the work of Kevin Shortcut. It's, to me, this is incredible innovation, yeah, leading the field forward. But you also need to understand, and Kevin Shortcut did, okay, he found this cryptic pocket, right? So what did he do? He didn't just target the pocket, because the pocket is cryptic. What does cryptic mean? It doesn't exist in 100% of the RAS molecules, right? So it exists for certain time. How much time? Depends on the stability of the conformation with that pocket. To overcome this, he used covalent bond, which stabilizes it. And of course, now we know many people follow it as well. Yeah, it's ingenious, yeah, this approach. So now people look for cryptic pockets, including us. But this is, yeah, but yeah.
Thank you.
Yeah. So I guess I should. Okay. So shall I remove this?
Yeah, well, I don't know.
Leave it.
Do you want the mouse?
Do you want the mouse? You don't care, so this way I'm sure I will not forget it.
Is this your coffee?
Still have coffee.
This is your copy. This is your copy.
Ruth, thank you so much for setting up the talk, and I appreciate your dedication and contribution to the space. My name is Carter Mitchell. I'm the CSO at Kemp Proteins. We've been around in one form or another for over 3 decades, making proteins in the Frederick, Maryland area. And the talk today, I mean, it's kind of hard sometimes to choose a talk title when you don't really know what you're going to talk about yet, but really it's looking at disordered regions and molecular modeling today, and to try to find as much information as possible to increase the likelihood of success in any sort of developmental workflow that we're trying to focus on. I apologize, I'm a mover and hand mover and waver and talker, so I'm going to try to keep myself leashed right here as best as I can. So I'm a structural biologist by training. I love it. I can't get over it. And actually, it happened when my mom asked me to transplant some, what are they called, crepe myrtles, when I was growing up in Texas. And it was super hot outside. We should never transplant trees when it's hot. It's stupid. I took a shovel, drove it into the ground, broke open a rock, and I discovered crystals. And oh my God, did it change my life. I then started getting all of these rocks and breaking them open. And I was just blown away by this idea of beautiful relationships, arrays, incredible geometries, right? I didn't care about diamonds and sapphires. I just loved the idea of things assembling in a regular array and building this type of beautiful scaffold that I just really enjoy. And then during my undergraduate career, I discovered crystallography. The fact that you can crystallize a protein, it blew my mind to take something so complex and to build this regular array into a crystal that you can see. And then you can fire X-rays at it, for God's sake. That's amazing. Right? So I want to go to a particle accelerator. I want to put a crystal on there, and I want to see the diffraction pattern. And the fact that you could discover a structure for the first time, nobody's seen it before. You see it 3 o'clock in the morning, drinking coffee all night long at Cornell, and you get to solve the structure for the first time. Like, that's amazing. It's really incredible. And this is a pictorial representation of that type of process that happens. It takes a long time. And to be honest, we can't do what we do today without the previous decades of research that the scientists before us have been able to build upon. So with the discovery of X-rays and the early 1900s into solving the structures of DNA in the 40s and 50s, right, into getting the Nobel Prize in '62 for pepsin. So this idea of macromolecular modeling is actually quite new, if you think about it, in the context of science in general. And then coupled with the new advances in computation, we're now at this kind of extremely fast cycle of incredible developments. But what the hell does it mean? And what is quality and what isn't quality? And when you look at the PDB, how many of those structures are actually generated in E. coli when in fact they're from humans or from some other sort of organism? And what is the story of the protein, the journey of maturation from start to finish, right? We start with, of course, the central dogma of molecular biology. We know that everybody here knows that that's kind of correct, right? You go from your DNA to RNA to our protein, and that protein is translocated or moved around in different places inside of the cell. It receives tokens in its journey to maturation, and those tokens change the shape and the structure of that protein. And we talk about disease states that are becoming phosphorylated. Well, those phosphorylations can actually change the dynamics and flexibilities of these dynamic disordered loops as a function of electrostatic charge, et cetera. So Kemp Proteins, we make proteins, obviously, and we use structure. So in this particular case, it's an example of a protein that was a reference protein for a clinical trial, and we use transmission electron microscopy for particle analysis, dynamic light scattering, all of that crap. So we're in the business of making proteins, and we care about structure. However, we actually start every program with thinking about protein structures. And if you guys have seen me talk before, I can't get over this particular slide. I love this concept of being able to take all of the sequences in the universe, well, they say the universe, but it's in the world, and then mapping them as nearest neighbor homologs. So it's a cluster analysis where all of these green nodes actually correspond to protein families, types with high sequence homology that's present there. And you can see that there's concentrations in certain areas. Oh, I can't show you anything with my mouse, I'm sorry. There are concentrated areas where it's very, very green, meaning that there's a lot of sequences in that space. And that means that there's a lot of potential information that's also known about those. So we can click on those nodes and we can find out about those protein families. so that we can come up with a strategy to be able to generate these types of proteins. But what do we do for the space in between the nodes where there's not a lot of information present? And, you know, we spent a lot of time trying to understand human proteins because it's directly related to us, right? We want to try to be able to solve disease and stuff like that. Can we take that information and translate it into another mammalian system? Probably. That makes plenty of sense, right? Is it exact? No, but it's pretty close to it. Can we take that information and translate it to a fungi? Maybe. Right? There's probably similarities and differences, so we just need to kind of understand how we would be able to do that. So what are the processes by which we come up with programs? And any structural biologist here or recombinant protein generation scientist would probably use some sort of format like this. You start with the sequence, you do as much literature research as possible to understand what's going on with that protein. You try to predict as much about that protein as possible so that you can identify what type of expression system you might want to use. And so we want to look at the subcellular localization. Is it going into the nucleus? Is it in the cytosol? Is it being secreted? Is it a membrane protein, right? We want to identify, is there a secretory tag? It's very important. Is it going to be secreted and then trapped in the membrane because of a transmembrane domain? And what types of post-translational modifications might be present on that particular protein of interest? On the right-hand side in this panel here, we've taken a protein that came from a metagenomic sample. We don't know the origins of the protein. It was in a soil sample, and we wanted to identify what type of path could we move forward with to be able to increase the likelihood of success of generating this protein molecule. We want to identify what kind of kingdom did it come from. We're looking at gene ontology. So what does it bind to? What is a potential predicted role of this protein? We also want to know, is it an enzyme or not? Because if it's going to cause interference with primary metabolism, we would like to come up with strategies that can protect that host so that we can increase the likelihood of success in generating the protein. And we look at the liabilities as a function of probabilities, right, on these charts in the middle. And anything that's above 0.5, we consider something that might be at risk. In the middle panel, it actually shows cell adhesion, it shows proteolysis, glycosylation positions, and the like, as well as a predicted secretory tag. So in this particular example, we have a native protein that doesn't have a secretory tag. Most likely a glycosylation is not going to be there, but we actually do have cell attachment potential motifs, which could be problematic for downstream processing. We generate a molecular model, right? And in this particular case, there was no AlphaFold model. And so this one is a Rosetta fold model that went through energy minimization using GROMACS, because I'm an energy minimization nerd. And what we find here is that if you look at that structural model and compare it with what the Raptor X right here, that plot right there looks at disorder propensity as a function of sequence. You can compare both of those together and kind of get an understanding of liabilities that might be present in the protein. And of course, we think about proteins as, well, biologists often think of them as circles, right? Structural biologists often think of them as cartoons, right? But the reality is these things are actually breathing. So biophysicists think of a moving target that's constantly changing, and Ruth elegantly highlighted Scheukett's work with accessing a cryptic pocket, right? That cryptic pocket is cryptic because it's moving. And so we shouldn't be thinking about static entities that are just moving throughout our cells. These things are always moving. There's a lot of energy inside of that. And also in the cytosol, what is it, 300 mg per mil of protein? So there's a lot of stuff going on in there, and it's very tightly compact. So you can understand or appreciate that there might be cooperative binding or some sort of fluctuations or dynamics of protein spaces that might be considered disordered. In this particular case, I say, hey, I'm going to put a tag on the C-terminus because I have structure there. And it's going to be probably pretty easy for me to generate this particular protein. So that's how Kemp will use, these types of tools to understand how to develop a program from a protein that's never existed before. But what about a protein that might be a little bit more disordered? This is another example of a more disordered protein. And you can see in the Raptor X plot there, that there are defined regions that have a higher likelihood of disorder, and that's a function of sequence, solvent accessibility. We're talking about sequence liabilities that are low complexity sequences, so like polyarch and serine and that type of stuff. You can understand that would have a lot of flexibility and fluctuations. We've also comparing here an AlphaFold model up on the top versus a Rosetta fold that went through energy minimization down below. And you can see, as a structural biologist, anybody in the crowd, the AlphaFold model is impossible. It doesn't really make any sense. There's just no way that a structure would look like that. So realistically, those loops are probably poorly defined in the structural databases. Like these are not involved in crystallographic packing interactions and therefore not resolved in crystal structure. So we have very low confidence in their atomic position as a function of the sequence and the protein specifically. But utilizing all of this information here, we can see that there's cell attachment. We can see that there's phosphorylations that are predicted over there. And actually, if you compare the phosphorylations with the disorder components, you can now start to tell a story where those components of the protein that are predicted to be disordered might actually have structure as a function of where it is in its journey inside of the cell at that moment in time. What I did here is I actually took the AlphaFold and Rosetta Fold models, superposed them. Global structural alignment failed, and you guys can see that we have these core domains, but in three-dimensional space, they're not necessarily organized in a way that we can superpose the entire structure. So I superposed the N-terminus. I got 0.35 angstrom RMSD, and you can see how this is kind of working. But what's more important to me is if you look at the right-hand side of this table right here, we are identifying proteins that are known to bind to specific regions, and those are actually coinciding with the disordered regions. So in this particular example for ABL protein, right, we know that it has a region that interacts with another protein. We have multiple areas where if you make a mutation in sites, you lose the affinity for a specific protein target, and that's corresponding to these areas right here. And so what I'm proposing is that utilizing a simple type of system such as RaptorX to identify disordered areas, and then looking back at the bioinformatic analysis and literature, you can actually start to tell a story as to why we think it's disordered, and it's a function of its natural evolution because it is involved in many binding partners as it goes through. So what does disorder mean, and how do we define disorder? And can we leverage a quick disorder prediction routines to find targets that need more consideration? And that's kind of my focus for the rest of the talk here. We evaluated the entire human proteome. It's about 20,654 sequences for disorder using RaptorX. We literally just did this, and it's just resulted in me asking even more questions. So I'm kind of excited about moving down further in this path, rabbit's hole of sorts. And RaptorX, just so you guys know, is a deep convolution neural fields. It predicts as secondary structure, solvent accessibility, and disordered regions. And the DISO is what we're specifically focusing on. Here's a couple of examples of disordered. So 100% disorder. AlphaFold is saying, that's probably going to form some helices. AlphaFold also on the middle panel is showing that there might be some beta sheets and stuff like that. And then down below, we have BCLF1, BCL2 associated transcription factor, and that is impossible, right? Anybody here can appreciate that we don't have a protein in our cytosol nucleus that is just a string, right? That's just all over the place. That guy is probably interacting with a lot of macromolecules. And I bet you that if we were to calculate electrostatic potential on the structure, that you would see discrete areas that have more basicity or acidity and those types of things. And so you can kind of weave a narrative that would explain what that protein would look like in real time. And so of those 20,654 sequences, we found that the lion's share of the proteins that have been described actually are kind of fitting into this bin of 0 to about 30% disorder. And that makes plenty of sense, because evolutionarily speaking, we're stating that structure might actually conserve components about the protein and keep it alive longer, right? It wouldn't be toxic to the system as we go through. I was really interested in the 100% disorder because it just doesn't make sense to me. And when I started just randomly selecting items, I kept on getting like 10 amino acids, 25 amino acids. I started asking, are these even proteins at this point? Like, what are we calling the human proteome? And you can see here, I went ahead and bend the average amino acids and standard deviations. And 100%, the amino acid average was 100, 105, and the standard deviation was 100. You can see that it starts to break down differently after that. So there were not a lot of entities that were 100% disorder that were very large. So because of that question about sequences, I also wanted to just see what is the average sequence length of proteins in the human proteome. And this doesn't necessarily take into account isoforms, okay? And what we can see here is on average, we're looking at about 200 to 400 amino acids. That makes a lot of sense to me. You could just start looking through these examples and probably prove it to yourself. But again, what is this idea about a 100% disordered? Down on the right-hand side, I took the biggest protein I could find that came from our binning analysis. It's only 715 amino acids, but the entire block is red. And you can see the alpha fold structure is just a bowl of spaghetti. And what does that protein do? It is a matrix protein. From my perspective, when I see matrix proteins, when I see cytoskeletal elements, when I'm cell adhesion, those types of things are problematic from an expression point of view. So how are you going to develop a model that's going to be able to depict what's going on here? Or how could you come up with a recombinant workflow that would be able to make this protein? 26% serines, 18% arginines. What a mess. Nobody wants to play with this protein. It's involved in doing things outside of the cell, right? And this particular example here in the 90 to 100% bin is mucin. I actually did not really appreciate that some proteins in the human proteome are so large that my computational system can't actually handle the full sequence. I have a maximum of 1,800 amino acids that I can put into our computation. So I have to split it up into 2 separate runs and then marriage these guys back together after the fact, which is kind of confusing. And if you want to do the right modeling, you probably want to break it into 3 so that you can have a model that bridges the 2 halves of the structure together. In this particular example, we're looking at 28 by 19 amino acid repeats. I have that box down below. So what we are finding is that the disorder seems to be coincident with low sequence complexity. That's not that surprising, right? But why did it evolve to have that capability? This is what we would like to discuss. This looks like I made a mistake, but in the back of this screen is actually the amino acid sequence to the largest protein in the human proteome. It's 8,384 amino acids. That is just mind-boggling enormous, right? I actually had to change the font size. It was hard to get it all on this one particular slide. And in this example, we're in the 50 to 40% disordered bin. We have a protein called mucin. If anybody's worked with mucoid-like protein extracts, they're the worst thing in the world. You put a drop of liquid in it, and the next thing you know, it turns into this viscous, disgusting, nasty, you can't put a pipette in there and take it out. It's terrible. And I suppose that mucin 19, I have not expressed this protein, nor do I ever want to. But I propose that it's probably going to be that type of disgusting, viscous mess as well. And we've seen a couple of instances as the mucins being some of the largest proteins that are in the database. But that led me to the other question. I started building a hypothesis stating, well, the larger the protein in size, the higher the likelihood of finding a disordered region. So the question I had was, are there any examples of extremely large proteins with 0 disorder? And that's what we found here. This particular protein is the CSMD2 protein with the cub and sushi domains. And if you guys know about sushi domains, they're often in complement. Recombinant factor H has the sushi domains and the like. And you can see that in this particular example, it's showing that it has a bunch of beads on a string type of domains down on the right-hand side. It actually took me a long time to fold this particular protein. And this is the example where I found out that my computational machine has a maximum of 1,800 amino acids, because you can see it actually just clips off there. It continues on even further. There is a transmembrane domain. I didn't have time to modify the slides to show this. So this protein is enormous, linear, flexible, yet they're stating it's not disordered, which I thought was really, really interesting. And so, in the sake of time here, we evaluated the human proteome for disorder using the RaptorX, right? Disorder seems to be coincident with a subset of protein types. Certainly the sequence, of course, is its function. And what we're going to do next is actually take all of these proteins and cluster them as a function of gene ontology so that we can have different motifs. And then I can ask a question. I'm looking for a DNA binding motif. What's the percent disorder? What do the models look like? What is their function? What structures are available to prove or disprove that we have a good understanding of what this sequence looks like in 3-dimensional space? After clustering those, the structural analysis will be completed. But really, this presentation serves as a call to structural biologists to focus on less characterized motifs and provide empirical data to improve our structural understanding. In the past, we've always clipped out the parts that were hard, right? I don't know if it's because we're lazy or I don't know, we don't have time or money or funding, right? Maybe it's because we were in the wrong host, and it actually would have been better if we were in a primary cell or a mammalian cell instead of HEK. I'm not quite sure. There's probably not a one-size-fits-all type of solution to the problem. But if we can better characterize the SH2 domain, in the ABL story, for instance, and have it interacting with its protein target or DNA, we can now gain a better understanding and have higher precision in our molecular modeling, which likely would improve the overall success rate of all of our discovery campaigns and actually decrease off-target effects, also decrease risks, and decrease the developmental time and money involved. I'd like to actually thank the Biotech Connector for the time, AlphaFold and RosettaFold for for providing the molecular components. Centauri is our molecular computation group. And then with this, I actually really love this slide. There is a message in there. Quality is in our DNA. You just need to use a methanotrophic bacteria to translate this. And that last bit, what can we help with, is actually translated there with a peptide that has the same amino acid sequence. So with that, I'm happy to open it up for any questions. Yes, sir.
Yeah, can you use, have you done anything looking at protein interaction, making predictions about what's going to bind to what and where?
What is going to bind to what and where, or I have two things that I know bind, and where are they going to bind?
Exactly, or do they bind to each other?
Yeah, so what we have been working with are fast calculations for protein-protein interactions. We found that the fast protein-protein interaction calculator allows us to identify areas that need more evaluation. We take that subset of information and then we go through a free energy perturbation, which uses something called alchem, which I disagree with and agree with. It's the fastest way we could do the computation at that time. And using FEP modulations, we're able to get very high accuracy in measuring how the proteins are interacting and where. And the energy involved with that, with the FEP basis simulation. There are groups that are developing monoclonal antibodies de novo. The CDRs are coming out of just an in silico type of computation, where they take an antigen and they're mapping a surface to that type of thing. So that's finding a de novo type of process. And of course, I'm sure you're aware of the previous protein-protein interaction types of softwares like AutoDoc, those types of things. Really, I think the problem that people have with the protein-protein interactions is that the amount of complexity in the computation is huge, because each one of those atomic positions has some range of movement. And I played around with the elastic network modeling and isotropic network modeling and seeing if we can perturb those systems as a function of energy. But that's using a simplified type of process with the network modeling. So I think that we're there, and computation-wise, we're finally at a point where it's cheap and cost-effective for us to do these types of calculations. But what I recommend is finding first something like Raptor X, the PPI calculation, and then after you find a cohort of your data that needs additional, really expensive computation, you go back through and do the high energy calculations there. That's what's been working for us to be able to bootstrap, some of our discovery and development efforts. And it's helped to improve affinities, thermal stability, and the like, because protein-protein interactions could also be a protein interacting with itself. Right, and that allows you to do thermal stability types of improvements and calculations. Yes, sir.
One question. So you've seen that you can split up proteins into pieces because of its size. How do you control for large scale effect? Because sometimes the large protein, you may have localized effects, you're missing the data effect that happen.
So I think it's very critical always to consider that these are merely hypotheses. We're trying to get some understanding of what it might look like inside of a solution. So you're absolutely right. And from my experience, when you look at protein structural modeling, there's usually 20 models that are satisfying the equations, and some of them have differences in their position. And so by looking at the 20 models as opposed to only the lowest energy model that's output, you're able to actually tell a little bit more of a story about the dynamics of a protein, potentially, right? And then also how they might fit together in the context of what we're talking about right here. And again, I just encountered this problem, so this is a potential solution, and I'd be happy to let you know what I find out after I piece these pieces together. Yes, sir.
It seems like you're right at the edge of where you really can't use X-ray or NMR because there's so much disorder. So have you tested your model with something that's intermediate where you know where there's several different configurations in solutions, say from multiple timescale NMR, and then see how does your model fit something like that?
Yeah, so I think that each of the structural tools that we have should be working together in concert for us to have an adequate story. With crystallography, we freeze out dynamics. And with Eta Ionith and cryopreservation and data collection at low temperature, we freeze out a lot of those vibrations. Room temperature X-ray diffraction actually has a lot of value. You can see a lot of information and actually understand electron, transport and those types of things as well. And then we look at NMR. Well, NMR has its limitations unless we're utilizing some fluorine-based methods, which makes a lot of sense. And that allows us to get this bridge, this time scale into, milliseconds, up to milliseconds, I believe, right? And then we have cryo-EM. The advantage there with the cryo-EM is that we're looking at many instances of the image, and we can actually calculate or fluctuations as a function of the amount of sampling that we've taken. So, and then of course we also have mass spec, right? And then we have small angle X-ray scattering. My point is taking all of it together and getting as much data as possible, and stop thinking about a static state of a protein, and understand that these are dynamic and very cooperative and interacting with partners. Proteins don't want to be by themselves. They're very social entities. They don't have a choice, to be honest. And they're standing shoulder to shoulder with partners throughout the cytosol, right? And so what we need to better understand is that this shapes the evolution of these proteins, and that we should stop thinking of them in a vacuum and think about it more in a crowded audience and that type of stuff, and understand the protein dynamics across all time scales possible is what I think should happen. And with the decrease in computation costs, I think we're really at this point where this is going to be taking off dramatically. All right. Thank you so much, everyone.
All right. See if I can run through this presentation very quickly in the last 2 minutes. So thank you so much for the organizers to allow me to present today, and thank you everyone for coming here bright and early breakfast time to come for this talk. So I guess the best way to start is right at the beginning. What is required to develop drugs? A lot of people say that, why is it taking so long? Well, the average time for taking drugs is maybe 10 to 15 years, and it costs $1 to $2 billion. And that's all for just one drug, because it's such a high clinical failure rate. And the question is, why is that? Drug development is very complex. There's a lot of processes you have to go through. You have to discover the target, you have to validate the target, you have to do hit identification, lead optimization, and so forth. And all of these things are, as you can see, there are a lot of different steps in this process. But the ultimate purpose is to reduce or remove the major causes of drug failures in this process. One, of course, the lack of exposure, the lack of efficacy, and also drug toxicity. And in order to minimize all of these things, there's a lot of research that has to go into place when you're developing a drug, so that when you actually introduce these drugs to patients, you have a much better confidence that the drug will work, is getting into the right tissues, and does not have unacceptable toxicities. So ultimately, when you are doing drug development, you want to have a good idea of what the ideal drug candidate should be, what we call the target product profile. What are the pharmacological properties of your drug? Is it highly specific, binding to your target, as Ruth had mentioned? What are the pharmacological properties? Does it have appropriate chemical properties? Is a drug stable? Is it pure? Does it have non-problematic synthesis? Synthesis is very important as well, because when you're doing drug development, obviously you want to be able to scale the molecule, and so if it has a very complex synthetic pathway, it may be very difficult to get it out to patients. The exposure is important, as well as safety and tox. And so an idea of what the overall profile is will help you identify the ideal drug candidate they can take forward into the clinic. And some of the ways to de-risk drug development is what we call the approaches of ADMET. So ADMET is basically absorption, distribution, metabolism, and excretion, as well as toxicity. And early in drug discovery, as very early stages, from hit to lead or lead optimization, you can evaluate a lot of your drugs for many of the properties that are important for having maximal exposure in the patient. And the ultimate purpose of these ADME or ADMET studies is to eliminate compounds that are duds before you take them further into downstream development. The drug should have drug-like properties, the appropriate solubility, permeability, and so forth. and these can be optimized in vitro. And the ultimate purpose of many of these in vitro assays is to mimic the in vivo situation before you introduce them to animals, before you introduce them to patients. And these are an example of a variety of different assays you can use. You can look at permeability assays. You can look at plasma protein binding assays. Permeability assesses whether the drug can get across the gut, for example, if it's an oral drug. If the drug is highly protein bound, then it'll be less available to be activated at the target or binding to your target. Metabolism, which would be the focus of my talk going forward, is also very important because if the drug is heavily metabolized very quickly, then it will have very poor exposure. But the metabolites themselves could have issues as well. They can cause toxicity or interactions with drug-drug interactions. And then, of course, the drug can be removed or is removed through the excretion processes, as well as looking at overall toxicity. So what do in vitro ADME assays assess? For example, aqueous solubility. Is a compound soluble? How hydrophobic is a compound? Will it be able to get across cell membranes? Is it stable through metabolism? Is it stable in the blood? And does it have any drug-drug interactions? And what's interesting about all of these studies for in vitro ADME is that they're almost all interrogated by mass spectrometry. So mass spectrometry is one of the standard approaches that in vitro ADME scientists use to evaluate, especially small molecule drugs. And very simply, there are a variety of, it's a whole topic in itself in mass spectrometry, but very simply, you have a way to introduce a sample, and the sample is then ionized through an ionization source, analyzed with a mass analyzer, and then detected. And for many of these small molecule studies, because you're using complex matrices, you use liquid chromatography to first separate the molecules from the matrix. You ionize it using electrospray ionization, which is a very mild way to ionize molecules. And then you go through a selection through these quadrupoles and then ultimately detect. And so in the LCMS triple quad setup, which is a typical setup you use for looking at small molecule drugs, you would have a mobile phase which basically goes through usually a C18 or some other type of column to separate the molecules as well as remove them from the matrix. And then through electrospray ionization, these molecules are ionized. And then you have 3 quadrupoles. And these quadrupoles are basically ways to separate and isolate the different ions that are produced during the ionization. So the first quadrupole or triple quadrupole setup isolates the major molecular ions, so the major species that is ionized. Then you have a second quadrupole, which is what the collision cell, which allows it to be further fragmented. And then the third quadrupole separates the fragments. And so in this process with a triple quadrupole setup, you can get a very precise and very clear understanding of the fragmentation patterns and the structure of a molecule. And one of the major uses of this type of technology is looking at metabolism. And so what is metabolism is essentially how does the body break down a drug? And this is important because, again, when you're doing drug development, if the drug is heavily metabolized or it's very unstable metabolically, it'll be removed from the circulation, it'll be excreted, and then it'll remove your potential efficacy. But also, metabolism can also be a concern because the metabolites, or may not be a concern, but you have to be interested in knowing what the metabolites are because the metabolites themselves may have pharmacological activity or potentially toxicity. And so metabolism usually happens, a variety of different organs can metabolize drugs, but especially if you're looking at oral drugs, using first pass metabolism or first pass effect is basically in the liver, is the major metabolic or major organ where metabolism occurs. And there's 2 major phases for metabolism, phase 1 and phase 2. And phase 1 is oxidative reductive or hydrolysis, and phase 2 is conjugative. And the purpose of these different phases for metabolism is basically to make the drug more polar so that it could be removed from the body because the body doesn't like foreign organisms or foreign species. Now, with metabolic stability assays, again, because the liver is a major organ for metabolism, use liver extracts, liver microsomes, which are basically ER fragments from the liver. liver S9 fragments or hepatocytes to evaluate the metabolic stability as well as looking at metabolite ID. So why is metabolism important? I mean, one, of course, is as I mentioned, the half-life of the drug. If it's metabolized, it can be removed. So you want to understand, what the metabolic stability is. But also, metabolic transformation is important because it gives you a sort of indication of potential toxicity. And this is a very classic example in the field. Of course, acetaminophen, Tylenol is metabolized by phase 2, glucuronidation and sulfation. But in a situation where you have very high dosing or have very high dosage, then it reverts to phase 1 metabolism, producing this NAPQI toxic intermediate that in very high levels can cause liver toxicity. And so by understanding the metabolic process, you can understand the fate of the molecule and what potential toxicities that can be anticipated. The other important consideration for understanding metabolism is looking at drug-drug interactions. And Ruth, as you mentioned, combination studies are very important for oncology and other types of diseases, but also many patients are on numerous regimens. They can be on statins, they can be on, oncology patients on average have, 7 different drugs they're taking simultaneously. So it's very important to understand whether your drug can have a drug-drug interaction, whether it's the parent drug itself or whether the metabolites can be involved in drug-drug interactions. And the major, again, the metabolizing enzymes, again, in the ER is the cytochrome P450 system. And so one of the things that we want to look at when you're looking at drug-drug interactions is whether your parent molecule or their metabolites can either inhibit these cytochrome P450s. So if you inhibit them, then you increase half-lives of concomitant drugs. If they induce the cytochrome P450 system, that allows for drugs to be easily eliminated, or if they're a substrate of cytochrome P450, so that the drug is actually processed. In addition, there's also transporters. Looking at P-glycoprotein and BCRP and others, but that's beyond the scope of this talk. But that's other important considerations for drug-drug interaction evaluation. And very, very recently, May of this year, the ICH, the International Council for Harmonization, had just released the M12 guidance, which gives a very nice guide to how to conduct these drug-drug interaction studies. And because it's a harmonized guidance, it combines the guidance from the FDA, the EMA, and other world bodies to come to a consensus of what are the appropriate studies so that we can really understand how to fully interrogate DDIs before they get into patients. And here's an example of an ICH-compliant phenotyping workflow. So as I mentioned, when you're looking at interactions with cytochrome P450s, one of the concerns is whether it induces cytochrome P450s, whether it inhibits, or if it's a substrate. If you want to understand how a drug is metabolized, first you would ID your metabolites, after mixing with liver extracts or hepatocytes. And this, of course, is done by LCMS to identify your metabolites. But then you analyze the metabolites and try to determine what type of changes have been undergone. If it's an oxidative, you test the standard CYP enzymes. If it's conjugative, you test the relevant enzymes, UGTs and others. And if your metabolites cannot be determined to be due to those processes, then you can interrogate further oxidative metabolites. And the ultimate purpose, of course, is to see which enzymes metabolize your drug or your drug metabolites. And that's important because when you're looking at drug-drug interactions, you have an understanding of what happens to the metabolites in the drug itself and which cytochrome P450s or relevant enzymes are going to be of importance when you want to look at drug-drug interactions. The other important consideration when you're looking at metabolism is the selection of non-clinical species. And that is also based on in vitro metabolism. And as I mentioned earlier, metabolism is very important because one is the half-life of the drug, but also the metabolites themselves can be very toxic. And in order to interrogate the toxicity, when you're developing drugs, you usually use two species, a rodent and a non-rodent. And one of the most critical aspects of evaluation in terms of drug safety is the identification of a pharmacologically relevant non-clinical species. If your species do not represent all the metabolites that are seen in human, then is not considered a relevant species. And so in this situation here, unfortunately, I don't have the pointer, but the rat and the non-rodent 2 can represent all the metabolites that you see in human, whereas the non-rodent 1 and non-rodent 3 have additional metabolites, and these are again are done in vitro using metabolite ID that are not representative of the human, and therefore they would not be relevant species for safety evaluation. So a case study that we had once was that there was a sponsor who wanted to perform GLP studies for their small molecule program. They had all the data that was necessary to take the drug into the clinic. They had efficacy studies, in vivo pharmacology studies that show the effect of the drug. They had PK studies that show how the drug is distributed and then excreted. And they also had MTD studies in rat and dogs, as well as 2-week tox studies. But what they lacked was in vitro metabolite data, and they hadn't done these studies. And it turned out when they actually did this study, the dog lacked a major human metabolite. And the problem with that is that major human metabolite, if it was present, could be a major source of toxicity. And so if the dog did not have that metabolite, then you cannot have a thorough evaluation of toxicity. So ultimately, they have to go back and repeat many of these studies with a relevant species. Yeah, what the relevant species is, it could be non-human primate, it could be anything. But the idea is that you want to make sure you understand the full metabolite profile and identify the relevant species before you go into a tox program, because then you can better predict clinical toxicity. So in conclusion, when you're developing drugs, you want to make sure you establish screening criteria to identify the appropriate drug candidate that has the best overall profile. The studies of metabolism are important not only for looking at half-life, but assessing metabolite toxicity, also identification of relevant species for safety assessment, and also very importantly to understand drug-drug interactions, because again, many patients are on a number of drugs that could potentially be problematic. And then finally, of course, mass spectrometry is an essential player in all of these studies, and it's particularly in ADMET studies, but all throughout drug development. So structural biology is something that is intrinsic to the drug development pathway. Thank you very much. Happy to take any questions.
So then you're recommending that we do in vitro metabolite analysis before larger animal studies, because otherwise it could be extremely risky. And in an effort to decrease the developmental time cycle, we can use this as a first kind of call of these drug compounds. Do you see that larger molecule workflows also require the in vitro metabolite analysis early on?
Yeah, so those are great questions. Typically speaking, we recommend doing the in vitro analyses for metabolite ID. A lot of times we do it in a number of species, usually 5 or 6, to ensure that you are capturing all the metabolites. Because if you do not, exactly what you're mentioning and what I mentioned earlier is that you may not represent the entire profile of molecules that may have a toxicity issue. So an early screen of metabolites across different species is very important and something you would definitely do, because we've seen situations where people have not done this, and then not only, I mean, one is regulatory, the FDA will not accept it. But second, in principle, it also would not be acceptable because those metabolites may be the major source of tox, which happens in many situations. In terms of larger molecules like biologics, they tend to be metabolized through conventional catabolic processes. So we don't go the typical metabolic ID. In many cases, unless they have some non-natural amino acid or something like that, we would have to be looking at metabolites. Generally speaking, we don't do metabolite ID because it's regular degradation through conventional biological processes.
And then since you guys are working on so many different programs, I assume, do you have any predictive tools that would classify a molecule or scaffold as having a high likelihood of being cytotoxic or failing the in vitro metabolite workflow? And you take your client programs and immediately put that through a predictor?
Yeah, so a lot of medicinal chemists have AI tools and a lot of in silico tools to predict whether the drugs have any liabilities in advance. But you still want to confirm them experimentally, just like you're doing in modeling. It makes great to do a model, but the experiment itself will demonstrate conclusively whether it's an issue. Great, thank you so much.
All right. Well, let's thank our great speakers one more time. Thank you so much, everyone. And I would also like to acknowledge our co-organizers of this event and the sponsors for the breakfast today. It's the Frederick County Chamber of Commerce, Mary and Kimberly. So thank you so much. So our next Biotech Connector event is going to be in November 21st, and the topic will be the next generation sequencing. So please stay tuned, and yeah, hopefully you will join us again in November. Thank you for coming. Grab some coffee. Thank you.